Fabulous. So I'm very excited, of course, to talk about the leptomeninges. My laboratory studies metastasis to the leptomeningeal space. And just to remind everyone and reorient you, remember the leptomeninges consist of the pia and the arachnoid, and they contain the circulating cerebrospinal fluid. They encase the entire CNS, and the spinal fluid that they contain is nutritionally quite sparse. And so they're kind of an anatomically isolated and nutritionally sparse environment. And it would seem pretty improbable that they would host metastatic disease, but of course they do, otherwise I wouldn't be here. When they do, we usually use the term leptomeningeal metastasis because any tumor can result in leptomeningeal metastasis, not just carcinomas. And in modern practice, the major contributors to leptomeningeal metastasis from solid tumors are breast, lung, and melanoma. Um, Unfortunately, the survival for these patients is quite grim, with an overall survival on the order of months. And so that just tells us that we have a lot of room for improvement. So one of the central paradigms in metastasis research comes to us from the Victorian pathologist Stephen Paget. And his famous seed and soil hypothesis, which I've written out here, has proven really durable as a metaphor because it captures this interplay between the intrinsic factors of the cancer cell and the microenvironmental selective pressure. I find this fascinating. However, when you visualize this in your head, you think about this, right here we have a bean seed in like this very loamy, delightful soil. And the thing is that I just finished telling you that the leptomeninges are a rotten place to live. And I think it's important to kind of remind ourselves that extreme microenvironments and extreme environments are capable of hosting life. Life finds a way. It's sort of the power of evolution. And so, this is the picture of kind of the sea floor, with all of the bacteria that can live there. So I think it's important to consider that soil that isn't congenial, to use Paget's term, can host cancer. And so one of the questions is, how do cancer cells adapt to the scarce nutrients that are around them? Or just rather than the loamy soil, visualize these plants in the desert. So I'm going to tell a couple of stories briefly today. One of them is the kind of the first story that came out of my laboratory, where we were it was our first stab at understanding this process. All pun intended, because we started with spinal fluid, right? So we took a look at spinal fluid collected from patients with newly diagnosed leptomeningeal metastasis from solid tumors, in this case, breast and lung cancers. And for these patients, we collected the spinal fluid and carried out 10x single-cell RNA sequencing. And what we found that was really cool was that, yes, we found cancer cells and immune cells like you'd expect. But the cancer cells clustered together, and that was surprising because that hadn't been seen in other data sets of metastatic disease, and that's something that hadn't been seen since in a lot of other metastatic sites. Usually, different tumors from different patients don't cluster together. And we thought that this reflected a conserved transcriptional response to this extreme microenvironment. So the selective pressure was so intense that it was forcing the cells to adopt a similar transcriptome. So when we tried to interrogate this data set, we thought about the selective pressure that these cells were under, and we thought about that irritating table that I showed you before that is from a lot of your kind of basic neuroscience textbooks. And we thought, what is the source of extreme pressure in this environment? And we thought iron is most limiting in this environment. And so we examined iron uptake in this space. So the postdoc that took this on, Yudan Chi, one of the things that she thought was that first you need to address the null hypothesis, right? Maybe the cancer cells don't really need iron, and so therefore it is not limiting for them. So she took a look at the levels of hepcidin, which is the hormone that's released from cells that are iron deficient, and transferrin, which is the canonical iron transporter that we all remember from basic physiology. And we found that the levels of hepcidin were elevated in the spinal fluid of patients who are harboring leptomeningeal metastasis, but the levels of transferrin were equivalent. So that told us that, yes, there was some iron deficiency in this space, but no, they weren't using transferrin to solve the problem. So we queried the data set looking for genes that encode products that are associated with iron binding and iron transport. And that's what you're seeing there in that heat map in A. And then in B, you can see that we found just two transcripts that were associated with iron binding and transport that were expressed exclusively by the cancer cells. Lipocalin-2, which is an iron binding protein, and SLC22A17, which is the lipocalin receptor. So we've got a nice pair here. Then the next question is really, what are they doing, right? So then we pivot over to our mouse models to be able to answer those questions of why and how and kind of more mechanistic questions. And we found that, of course, first, we found that the mice have disease that's quite similar to that we see in humans. So you can see this is a C57 black 6 mouse hosting cancer in its leptomeningeal space. When we do multicolor flow cytometry, you can see the cancer cells there in orange. And when we collect spinal fluid from either the mice or patients, we find elevated levels of lipocalin-2. So that tells us that, we're on the right track, at least in terms of the mouse models. Then to determine if lipocalin-2 was important for the pathophysiology, we knocked out lipocalin-2 expression from the cancer cells and found that inhibited growth in the space. And that's what you're seeing there in A and B. And then in the row below, what we're showing here is that we took cells that didn't have any real propensity to grow in the leptomeningeal space and didn't express lipocalin-2. And when we force those cells to express lipocalin-2, this accelerates their growth in the space and accelerates the death of the mice. So this plus many other experiments, including an experiment here where we're measuring iron levels in the cellular material that we collected from the mice, led us to a pathway whereby inflammatory signaling from the microenvironment was leading the cancer cells to be able to generate lipocalin-2 and SLC22A17, and that those gene products were allowing them to outcompete macrophage in the microenvironment for this limiting iron, this scarce resource. So because the cells were all fighting over the same limiting reagent of iron, we reasoned that what we needed to do was to eliminate the iron from the space. We needed to chelate away the iron, so then that would inhibit the growth of the cells. So to do that, we used the drug desferoxamine, which is iron chelator in practice, and we gave it in a new direction. So rather than giving it either by mouth or IV, which are the standard routes of delivery, we gave it intrathecally to the mice. And when we did, either early in the disease course or late in the disease course, we found that inhibited cancer growth in the leptomeningeal space. And chelating copper with D-penicillamine, that's the line in gray, didn't make much of a difference. Okay, so these data, and of course many others, prompted us to be able to open up a clinical trial of a phase 1 clinical trial of desferoxamine for patients with solid tumor leptomeningeal metastasis. The trial is still open and accruing. I can tell you that the phase 1A trial, phase 1A portion is complete. So we've found a dose. It's well tolerated, and we're even seeing an effect in terms of an effect in terms of the anti-tumor properties, which is really exciting. So if you have patients that have leptomeningeal metastasis and you're looking for a clinical trial, I know there are not many, this might be an option for you. Feel free to contact me. So the meat of the talk today is about a related story, which is this same idea of how do cancer cells respond to this microenvironment? Or as one of the people that was listening to a talk that I gave asked, he said, how do the cancer cells know they're in the leptomeningeal space? And at the time, I thought that was such an irritating question, and now I think it's a really cool question. So Q&A is the best part of any talk. So to answer this question, the graduate student in my laboratory, Shinran, who took this on, she reasoned that, okay, if the cancer cells, these leptomeningeal metastatic cancer cells, we call them lepto-M cells, if they can grow in the leptomeninges and they can grow elsewhere if we implant them there, we can use those as a tool. We know that they have the capacity to grow in the leptomeninges and elsewhere. Why don't we use them as a biological probe? And she reasoned that probably there would be some epigenetic change because this is a big change that these cells are encountering. So she took the cells and she implanted them in either the leptomeningeal space or in the primary site. So in the case of LLC in Lewis lung carcinoma, that would be into the lung, or in 4T1 model into the mammary fat pad, the mouse breast. And then she harvested the cells and carried out ATAC-seq. And working with Richard Koch and PJ Hamrad, we were able to find that the chromatin accessibility of these cells are really clusters based on the microenvironment that they find themselves in. It's not a cell intrinsic property. It's really based on a signal that they're receiving from the place that they are in. When we did network enrichment analysis, we were delighted to find all kinds of developmental pathways that were upregulated. It's crazy. So many. Look at this. It's really hard to find something that's not a developmental pathway here. So we decided to pursue this angle, and we started to look at the chromatin landscapes, reasoning that we wanted to really focus on areas of open chromatin that were conserved between these two mouse models in the leptomeninges, regardless of whether the cells were adherent to the leptomeningeal space or floating in the spinal fluid. And so that would, of course, be group 7 here. And when we look at group 7, we find a motif analysis revealing that this retinoid X receptor is enriched in this space. And moreover, you can see that it's enriched more strongly in the adherent population than the floating population. So keep that idea in your head for later. We took a look at that at the level of transcription, of course, because if there's open chromatin, you would then expect there to be transcription downstream of relevant start sites. And we do indeed see transcription downstream of RXR, LXR motifs in these populations. So, why was this exciting to us? It's because RXR is, of course, the retinoic acid receptor, and retinoids play these really important roles in nervous system development and homeostasis. I know you remember this from all of your prior school. However, I'll remind you that the retinoid signaling is a two-cell system, right? So we consume vitamin A, we take up vitamin A, and retinol gets hooked up to a carrier protein called RBP4 in the liver, where it can circulate. And retinol bound to RBP4 is then taken up by the first cell through STRA6, where it undergoes a series of dehydrogenase reactions in order to be able to generate retinoic acid. Retinoic acid is a freely diffusible, nice and greasy molecule. Unfortunately, it's has a very low half-life. It's a pretty evanescent molecule, and it'll diffuse into the next cell. And that next cell, our retinoic acid is then able to bind to its nuclear receptors, RXR or RAR, plus a co-receptor, and then can initiate signaling. So this is something we've seen all over the place in the nervous system, right? So classically, you think about mesodermal cells generating retinoic acid to signal to the neural plate in really, really early embryology. You think about the hippocampal meninges generating retinoic acid to signal to the dentate gyrus neurons, Schwann cells signaling to neuronal cells in the periphery, right? So we measured retinol, of course, in the spinal fluid. This was really difficult. So we had to work with our friends up in Boston, the Namakanorx lab, and we were able to find elevated levels of retinol in the spinal fluid of both patients and our mice in the setting of leptomeningeal metastasis, which was pretty cool. And so then we realized we have to solve a problem here, right? We've got to figure out who's generating the retinoic acid, so who's my generator, and then who's my receiver in the CSF. So the generator, that's going to be the one that's going to take up the retinol and generate retinoic acid through all of those dehydrogenase reactions. And then the receiver cell, well, that's probably the cancer cell, right? So we have to figure out what the cancer cell is getting out of this relationship. So for the first part of the question, we started thinking about the PL cells. So remember that PL cells are descendants of neural crest, right, in the forebrain, and direct descendants of mesenchyme in the midbrain and hindbrain. And so both of these cell types are known retinoic acid producers in the leptomeningeal space. And so it seemed like they might provide a nice local source of retinoic acid. And indeed they do. So when we stain up our leptomeninges in our mice, you can see that in the presence of leptomeningeal metastasis, you can see that these mouse leptomeningeal fibroblasts are expressing RALDH1 and RALDH2. You can also see that that's kind of a mess in there. You can see that those leptomeninges become quite raggedy, even in a pretty well-controlled mouse model. The same thing is true in the case of human meninges in our autopsy specimens. We can see elevated levels of RALDH in the meninges of patients who have passed away from leptomeningeal metastasis. So what might be downstream of retinoic acid? Well, remember, we were really interested in RXR, LXR signaling based on our ATAC-seq. So we thought about other physiologic contexts where we've seen RXR and LXR signaling, and that of course would be within the liver, where this signaling leads to de novo fatty acid biosynthesis, cholesterol efflux, and cholesterol homeostasis. And so we investigated that possibility. So Shinran very cleverly generated a system where she was able to recapitulate the microenvironment of the leptomeninges. So you can see here, this is an in vitro system with just cancer cells. And on the left is the Lewis lung carcinoma model, so the lung cancer model, and then on the right is the B16 melanoma model. And what you can see here in this heat map, these are the genes that are associated with de novo fatty acid biosynthesis, FAAN, ACACA, SCDs, and SREBPF1. And what's really cool, actually, is that retinol itself, so remember that's the precursor to retinoic acid. If we give the cells retinol, we don't see any change in their transcription of these genes. It's really just retinoic acid that's able to induce this response, either all-trans retinoic acid or 9-cis retinoic acid. So that's really speaking to the fact that these cells need a friend, right? They need the help of a generator of retinoic acid in order to be able to induce transcription of these genes. We find evidence for this same level of signaling in our human data set. So the single cell RNA-seq data set that I introduced to you earlier, we requeried it, and we found elevations in all of those RBPs and in ACACA, FASN, and SCD, really telling us that the cancer cells in the human context are also receiving these signals. We wanted to double check to see if the cancer cells were really really taking up retinol themselves, or whether they really were relying more on retinoic acid. So we knocked out STRA6, which is that the gene product that's able to take up retinol, and we found that that didn't alter the growth of cancer cells in the leptomeningeal space. However, FASN, the gene product that's associated with de novo fatty acid biosynthesis, that does, when you knock out FASN, you inhibit cell growth in the leptomeningeal space, and that makes sense, especially knowing that there's very little available lipid in the leptomeningeal microenvironment. So Shinran thought, this is good. However, we found all of this information by looking originally at retinoic acid receptor, right? So she thought, what happens if I knock out the retinoic acid receptor or the co-receptor in this system, LXR? And so this is actually kind of a bear of a project, because there are 3 retinoic acid receptors, alpha, beta, and gamma. And then, of course, there are 2 LXR receptors, alpha and beta. And so she did a double knockout of the LXR receptors and a triple knockout of the RXR receptors. And to our delight, found exactly what you see here, that in the absence of the retinoic acid receptors, the cells are no longer competent to be able to do de novo fatty acid biosynthesis. So when we take these cells and we put them in a mouse, we find something even better. So of course, when you knock out LXR, we see a little bit of an effect. That makes sense. But when you knock out retinoic acid receptor, you see a really profound effect on the cancer cell's ability to grow in the space. And we see something we've never seen before, which is survivors. Even in my iron chelation studies, we always saw that the mice eventually died from their disease. In this case, we have long-term survivors, which was pretty cool. So we wanted to know what the result was downstream of this. And so what Shinran did is she then carried out metabolomics of the spinal fluid of these mice, and found that knocking out RXR inhibits production of these FASN products, which is really satisfying because that's what you'd expect. However, this was, in vitro. So to answer this question in vivo, we carried out spatial metabolomics on brains that we had isolated from the mice. Now, I'll kind of walk you through this briefly. So you can see here we've implanted the cells in the lung, just like we did at the very beginning of this story, or in the brain, in the leptomeninges. And then we first carried out, the second row was looking at glutathione. Glutathione is going to help us see where the tumor cells are. And you can see there's a nice hunk of it in the lung, and you can see it there in the basilar meninges in these first two. I don't know if you can see my mouse, but in these first two, and in the second two in the next row, you can also see in the basilar meninges a nice little coating of glutathione. Phosphatidylserine is going to mark the white matter for us, and you can see some nice white matter tracks being highlighted in the next row down. And then in the third row down, you can see some unbiased segmentation where we were looking to see if the cancer cells had their own metabolic response to the leptomeningeal space, and they do. Finally, we were able to see that the cancer cells in the leptomeninges are making more products of FAAN. So we see more palmitic acid, stearic acid, oleic acid, and these are products of well, at least palmitic acid and stearic acid are products of FAAN. And then we see, of course, other products of lipid metabolism in the space. I wish we could have done the same experiment with RXR knockouts, but of course, the RXR knockout doesn't have any tumor in its space, so that makes it quite difficult for us to carry out spatial metabolomics when there's no tumor there. So in sum, what I've shown you in this second story is this idea that retinol in the spinal fluid is then taken up by the leptomeninges, and that the leptomeninges, these leptomeningeal fibroblasts, become a local source of retinoic acid. That retinoic acid then is able to signal to the local cancer cells, and that these cancer cells then are able to metabolically reprogram themselves and become competent to be able to generate their own lipids through de novo fatty acid biosynthesis, but also, data not shown because of time, cholesterol biosynthesis, as well as lipoprotein biosynthesis and phospholipid metabolism. So I think in general, what I would hope for you to receive from this sort of talk is this idea that yes, cancer cells are receiving and are receptive to sort of a wide variety of microenvironmental signals, and that these microenvironmental signals, whether inflammatory or developmental or potentially even neurotransmitters, right? All of these signals are going to enable the cell to be able to overcome some of the constraints of the leptomeningeal microenvironment. An open question, of course, is how might the cancer cells influence brain function, right? So how, if all the arrows are going in one direction, what's happening? What is the molecular call-in response in the other direction? So with that, I wanted to acknowledge all the people who did this work, my really terrific laboratory. We all love CSF, if you can't tell. We work with a lot of spinal fluid, and we have the best time. And they are just lovely, dedicated people. I also wanted to thank, of course, my collaborators who I've given shout outs to along the way, my support, because funding for science is not easy, but most importantly, my patients and their families. They are some of the most altruistic, generous people that I've ever met. They are facing what is arguably one of the worst diagnoses in medicine. And then they still come and see us in clinic. They come in, they ask for more ways that they can help. They donate spinal fluid. It's really remarkable. Thanks so much.
Thank you very much. That's such beautiful work, and it's so exciting to see finally more understanding about what's going in this, like what the physiology is of the leptomeningeal compartment. Would you talk a little bit about, is there a challenge in sort of potential escape mechanisms in treating the leptomeningeal space for things like nodular growth that might have their own little microenvironments that might differ?
Yeah, I think that's a really terrific point. And it's actually a little bit underlies how Shinran designed her experiment. So when we collected these two sets of data separately, we were thinking about this question. We thought, there might be something very different about the cells that are adherent versus the cells that are floating. The story I told was the first pass where we were looking to see what was conserved between the two of them. But there's a really terrific student in my lab. His name is Clark, and he's working on this question, this exact question. So what is the difference between the floating and the adherent? is there an equilibrium between them? We think there is. And when we have this equilibrium between floating and adherent, what are the differences? Like, in order to target the adherent population, do we need to target something different in terms of metabolic kind of qualities? Or maybe those are kind of more indolent cells, in which case should we try and just get all the floaters to kind of stick down and then radiate them? Who knows? But yeah, we're really interested.
Thank you very much. Thank you for joining us.
Thanks so much for the invitation. It was a lot of fun.